Hello, welcome you all for the family of our BioGoogle. So in today's video, we will discuss about the types of DNA based on their occurrence, means in which organism they are occurring. On that basis there are few types of DNAs. Okay. Firstly, we all know that when we say the word organism, we can find two types of organisms. One is prokaryotic organism, other one is a eukaryotic organism. Okay. So the type of DNA is different in prokaryotes as well as different in eukaryotes. Right. So we'll see how the DNA is there in prokaryotes as well as there in our eukaryotes. Okay. We'll see two types of DNA in prokaryotes and eukaryotes. Right. Firstly, first category, prokaryotic. Better we should know eukaryotic first to understand better. Otherwise we will be in a confusion. First we will see the eukaryotic DNA. Eukaryotic DNAs. Okay. Again, eukaryotic, we can find two types of DNA. Okay. Here, the first type of DNA is nuclear DNA. As we all know that in plant cell as well as animal cell, we have a nucleus. So the nucleus has the nucleolus and chromatin fibers or a chromatin network. So this chromatin is made of the DNA. It may be heterochromatin or euchromatin, but both of them contain the DNA. This DNA present in the nucleus is known as nuclear DNA. Present in nucleus. Okay, and seen in plants and animals. Higher plants and higher animals. Coming to the type of DNA, it is linear DNA. There is another word called circular. Linear means the two ends of the DNA are free. One end we can call it as 5 prime, other we can call 3 prime. One end here we can call 3 prime, other one as 5 prime. Just like this, the two ends are free. That is linear DNA. Circular means the DNA, though it is double stranded, it is joined each other. Means the ends are not free. But here the ends are free. Okay. So it is even double stranded in prokaryotes, but it is circular. But here it is linear. Okay. So in eukaryotic DNA, it is linear type of DNA. It means where we can find the free ends. Okay, but in circular we cannot find the free ends. Both the ends of the DNA are fused to each other. That is known as the nuclear DNA. Present in the nucleus. Remember, present in the nucleus of the plant cells, higher plant cells as well as higher animal cells. Sometimes even protists, amoeba, euglena, paramecium, slime molds, protozoans all have the nuclear DNA. Okay, but when we come to the cell, especially the plant cell, we have organelles called mitochondria and chloroplast. Those organelles also contain a DNA. Okay, that DNA is known as is known as organellar DNA. Organellar DNA means DNA present in the cell organelles. DNA present in cell organelles. Okay, so present in cell organelles. For example, we have mitochondria here. We have a chloroplast here. So the mitochondria and chloroplast, they have some sort of DNA. They have some sort of DNA. This DNA is known as organellar DNA. Nothing but the DNA present in the cell organelles. Okay. So seen in mitochondria and seen in chloroplast. Okay, so the DNA present in mitochondria is known as empty DNA, and the DNA present in the chloroplast is known as plastidome. So DNA present in mitochondria is known as empty DNA. DNA present in the chloroplast is known as plastidome. Okay, because of the DNA, these two organelles, mitochondria and chloroplast, these two are known as semi-autonomous cell organelles. Just because they have a DNA in them, they are known as semi-autonomous cell organelles. What is this? Okay, when we say the word autonomous, it means it is not depending on anyone. It is not depending on anyone. Means not depending on any organelles. Okay, not depending on DNA. Okay, but with this DNA, the mitochondria and chloroplast, they can synthesize about 70% of the proteins, because the DNA becomes mRNA becomes protein by transcription, translation. So with the help of this DNAs, the mitochondria and plastidome, the mitochondria and chloroplast can synthesize about 70% of the proteins. But for other 30% of the proteins, cannot be synthesized by the mitochondria and chloroplast. For this purpose, they are depending on the nuclear DNA. Hence they are called semi-autonomous. Okay, if they could synthesize 100% of the DNA proteins, they could be known as autonomous cell organelles, because they are not at all depending on nuclear DNA. But they can synthesize, with the help of that DNA, they can synthesize only 70% of the proteins. But for the other 30% of the proteins, they have to depend on the nucleus. Nucleus means nuclear DNA. Hence the two organelles are known as semi-autonomous cell organelles. Okay, just because they have DNA in them. Okay, so they are known as, therefore, chloroplast and mitochondria are known as semi-autonomous cell organelles. Semi-autonomous cell. Remember this point. Okay, this may help you in a NEET and theory examinations. Okay, you should be knowing what is nuclear DNA present in nucleus, what is organelle DNA present in the cell organelles like mitochondria and chloroplast. Right? So these are the two types of eukaryotic DNA. These are the two types of eukaryotic DNA. Okay, now we'll see the other type, that is the prokaryotic DNA. We'll see other two types, they are prokaryotic DNA. Right. So the second type, prokaryotic DNA. Okay, so when we say the word prokaryotic, we have bacteria, we have mycoplasma, we have cyanobacteria called blue green algae, or we have rickettsia, or we have polychaetes, we have polychaetes, etc. So in those organisms the DNA is different, completely away from the eukaryotic DNA. So we will look at those types of DNA present in prokaryotic cells. So the first type of DNA is chromosomal DNA. Chromosomal DNA. Okay, so as the name indicates, the chromosome which represents the the DNA which forms the chromosome or which helps in the transmission of characters from one generation to other generation is known as a chromosomal DNA. Okay, so single DNA present as chromosomes. Okay, and it helps in heredity, that is transfer of the characters from parents to the offsprings. Transfer of characters from parents to offsprings, and it will be responsible for only normal activities or functions. For example, normally birth, growth, development, reproduction, finish. Other than that, this cannot perform any other work. It cannot perform any other work. Okay, we'll draw a small bacteria to understand it. So here we have a circular DNA, right? Circular DNA, right? Here this circular DNA is known as nucleoid. Remember the word in prokaryotes. In bacteria, the chromosomal DNA is known as the nucleoid. Chromosomal DNA is known as nucleoid. But here the nucleoid is the association of three components. One is DNA, next one is RNA, and some of the polyamines. DNA, RNA, and some of the polyamines. All together they will form the nucleoid or the chromosomal DNA. Right. So this chromosomal DNA is responsible for only the normal activities, normal functions or the basic functions of the bacteria or the prokaryote. Nucleoid. Okay. The second type is very important. The second type of the prokaryotic DNA is very important. That is extra chromosomal DNA. Extra chromosomal DNA. Okay. Extra, additional, extra functions. Okay. It's not normal DNA. It is extra DNA. Okay, whatever we have seen earlier that performing normal functions, this DNA will perform additional or extra functions. Okay, so in addition to chromosomal DNA, there is a additional DNA or it is circular DNA. Circular DNA, and it is self-replicating also. It is self-replicating also. What is this? Circular, self-replicating. Yes, circular means as it is the circular DNA, round. The two ends are fused. Okay. Self-replicating means when one copy of the DNA forms two copies. The process is called replication. The process is called replication. The process is known as replication. So, this DNA will replicate, but it will not depend on the chromosomal DNA for replication. It will never depend on the DNA, chromosomal DNA for replication. It will replicate itself on its own and it will express its characters on its own. It will not depend on a chromosomal DNA. So this is known as plasmid. This is known as a plasmid. Okay, remember this word plasmid, very important for your examination, very important for the examination. So this is the plasmid here. But in various types of prokaryotes, there are various types of plasmids. There are various types of plasmids. Each type of plasmid perform individual function or a specific function. Okay, depending on the type of the function, the plasmids are given various names. Okay, I have told already this chromosomal DNA perform normal functions. This performs additional functions. Or whenever the conditions are abnormal. In such cases, this will help. Okay, I will write few examples of the plasmids performing various functions. Okay, so plasmid. The plasmid is known as Ti plasmid. The plasmid is known as Ti plasmid. That is tumor inducing. The plasmid is known as Ri plasmid. Ri stands for root inducing. Or it's also known as R plasmid. That is a resistance factor. Means that gives more resistance against the antibiotics. Or F plasmid, or it is giving fertility factor. It is giving a fertility factor. Okay, so Ti plasmid for tumor inducing, Ri plasmid for root inducing, R for resistance factor, then F for fertility factor. Sometimes the plasmid is also used as vector in recombinant DNA technology. This plasmid is also used as a vector in recombinant DNA technology, in biotechnology. We will study this again detail in biotechnology, first chapter and second chapter, means 11th and 12th chapter in your second PUC. Okay, like that there are various types of plasmids performing various functions. So these are the plasmids and these are the various functions performed by the plasmids, respective plasmids. So this is the extra chromosomal DNA known as plasmid. You may have one more question, what is plasmid? The answer would be the self-replicating extra chromosomal DNA present in the prokaryotes, or circular self-replicating extra chromosomal DNA known as a plasmid. Okay, which helps the bacteria whenever the conditions are unfavorable. Otherwise, the DNA, chromosomal DNA will be performing normal functions. But in cases of unfavorable situations like high temperature or low temperature or any other crisis, environmental variations, this DNA will help. Okay, I think this is enough for this video. Okay, in further videos we will have a discussion on other concepts also. Okay, means like structure of DNA or the Watson and Crick model of DNA. We'll see in further classes. Okay, if you really like the video, if you like the explanation, if you want to share, of course you can share the video with your friends, with your students, etc. And if you are visiting the channel for the first time, please press on that red colored button, subscribe button. There is no loss if you press on that. Okay, the sad side is people just watch the video, make use of, but they will not subscribe to channel. Okay, you are not paying for that channel, it is a free. But just the thing is, you just have to press that bell icon after subscribing. Okay, so it's my kind suggestion. Follow the channel for other chapters. Right, of course I will be uploading the videos in everyday basis, but if you want any more specific videos on a particular day, if you are preparing for any exam, if you have any more doubts regarding any concepts in biology, you can just add a comment over the YouTube in any video. I can come back to you soon with that particular video which you want. Okay, till that, once again rewind the video. If you got any more doubts in this class, you please write a comment. I will be definitely get back to you either in the comment section or in the next video. Okay, for further updates, please keep following our channel. And one more thing, we have a Facebook page also. The page name is BioGoogle, the same BioGoogle. Okay, the YouTube channel name is BioGoogle, the Facebook page name is BioGoogle. Please follow the channel also. Okay, we are uploading the many such biological information in the Facebook page. So better you follow the Facebook page BioGoogle, so that you will get many more educational updates or educational information regarding that, etc. Right? So hope this is enough for today. We'll see you in the next video. Till that, keep learning. Thank you. Bye bye.